in this video, we are going to learn about the retrieval of a gene from NCBI. NCBI, as you all know, NCBI stands for National Center for Biotechnology Information, which is a composite database and is kind of one-stop shop for most of our bioinformatics needs. So if we want to retrieve, in this drop down menu here, so these are the names of the various databases that can be accessed, and this is the search bar, so where we can type our query, and then we will get the hits. For example, if if I say type I want to know how much information is available in NCBI database regarding a keyword, say human insulin. Then I will type human insulin here and I will hit the search button. So by doing this, I am going to get all the hits, all the information. So basically, related to human insulin, there is a lot of information available in 29 databases, like in literature, in genes, and in proteins. And in these literature also there are various subsections, and in front of each subsection the number of hits that we have got have been mentioned. So this is when we perform the search without selecting any database. So it returns us all the information present in the databases. Right now here PubChem and all is also there. Now when we want to perform, so this is a very general vague search. Now if we want to look out for, say, for a gene, say FAD2 gene in Arabidopsis thaliana, for example. Gene is a nucleotide sequence, so either we can select the nucleotide, or we can straight away select the gene database from the drop down menu, and we can type here FAD2 gene in Arabidopsis, and we hit the search button. So by doing this, we have got the various results. So the search has been performed only in the gene database, as a result of which we have been returned 8 results are showing, because now our query has got FAD2 and Arabidopsis also. So basically all those entries which have got FAD term in them and also the Arabidopsis thaliana have been returned, right? So therefore now, but we know we were looking for FAD2 gene, so we click on to this FAD2 gene, which stands for fatty acid desaturase 2, present on the chromosome number 3, and this is the accession number, and this is the stretch of the chromosome on which this gene is present, and these are the various names of this FAD2 gene. So we click on to this FAD2 gene, and that's how we've been directed to this page. Now whenever we are retrieving a gene sequence, okay, that goes without saying that we have to retrieve the sequence always in the FASTA format. So basically through this gene database page, we get to know about the various aspects of the gene, location on the chromosome, on the genomic sequence. So here we see these hyperlinks. So we click onto the GenBank if we want to go to the GenBank page, or we can directly click onto the FASTA, and that's how we go to the FASTA format of this particular sequence, right? So this is the sequence that we have got. Now since this gene is present on the chromosome 3 sequence, so therefore only that part of the chromosome has been shown here which contains the information or the sequence related to the FAD2 gene, and therefore as you can see in this right hand side top window, that there it is, the selected region has been shown here. So if we click on the whole sequence, then and we hit update view, then this entire view is going to get updated, and we are going to see the entire whole sequence of the chromosome 3, but which is not the case. So that's how it is. So in order to save this, the faster format of the sequence is always it is saved in Notepad, because Notepad is the tool that does not change the formatting of the sequence. So we copy the sequence and we go to the Notepad straight away. We open the Notepad and we press the sequence and we save the file by giving it an appropriate name, say FAT2 gene 80 stands for Arabidopsis thaliana, and we save it on the desktop. So this is how we can retrieve the sequence of the gene. And now we can go and see this sequence. So it is present here. So when we click onto this, so this sequence opens up. Yeah, so